Hi, Frank. How are you? Nice to see you.
Yeah, I just need to plug in my headphones.
Hi,
Or
Victoria. Oh, it's so nice. We have a group together. Makes me happy. Thank you. Hi, Z. Hi, Anna. Hi, Manas. How are you? Hey, Z.
Hey, I just wanted to say hello. It's raining here in Western Mass just for a minute, so I'm really happy I'm outside taking photos and talking to the birds because we're in a severe drought. So every time we have rain here, we're just so happy. But I wanted to say hi and just say thanks. And yeah, I love this. I love your club and all the work you guys do. And yeah, it's I'm glad that I bounced in here. So thanks, Katerina, Frank, and Victoria, and welcome to everybody else. Oh, hi, Oza. Welcome to Clubhouse and one of the best clubs on Clubhouse. I'm done speaking. Bye.
Thanks, Z. You're always so nice. I really appreciate it. Can everyone hear me fine? In between, I have the red
you're
bar.
absolutely fine.
Okay. I
Crystal
don't know.
clear.
That's so nice to hear, Z. Thank you. That's really nice. I think it will be a really interesting room. Let me put up the organization website where our guest speaker works for. So in the meantime, we have something we can check out. Let me put up the link.
I was thinking about the topics. What do you think? What topics should we set?
Yeah, I think there's no option like evolution or something like that. It's just biology, science, and I don't know. I can't think of. Can you think of? They don't have too much available.
Oh, by the way, I think there's some leverage of this club to make Clubhouse add some things that we could use. So I think you could ask, Clubhouse to add something that you need.
Yeah, I think Victoria tried it for us, but so far not very successfully. Oh, I wanted to. Hi, Namanu. How are you?
Hi,
Namanu,
hi. I'm great. I'm great.
thanks for
Uh,
coming.
yeah, you're welcome. It's a pleasure to be here. Yeah.
Yeah, no,
So it's on evolution and evolution of form blood animals. Okay.
yeah. I wanted to introduce you to Frank, Victoria, and Z, and everyone. Namanu asked about Science Society and how he could help. So, I said we can join some rooms and then see how he do things, and then maybe suggest based on his backgrounds, like topics, guest speakers, and so on. So, yeah, welcome.
Yeah. Thank you. Hi, Frank. Hi, Victoria. Hi, Zed. Nice meeting you. And yeah, I'll just be here and listen to the discussion. And yeah, I'll hope to join you or propose some rooms like based on my background or something in the cutting edge field of my in my field especially. Yeah, so I'm here.
Yeah, thank you. We will start in around 5 minutes. Our guest speaker will be here shortly. So, in the meantime, please check out the organization website, Paleomos. It's this organization promotes scientific development in Mozambique, Africa, through the preservation and enrichment of its paleontological heritage. And the FCT, which is the Foundation for Science and Technology in Portugal, supports this organization, and our guest speaker Ricardo, he is at the University of Lisbon, but yeah, he works and does research for this for this organization. So feel free to check it out in the meantime.
Obrigado.
Hi, Ricardo. Olá.
Hello.
How are
Boa
you?
tarde.
Boa tarde.
All good.
We are the only Portuguese speakers, but Victoria here, she almost Portuguese now, but she can't speak right now. I'm sorry. She's traveling, but she's here listening. How are you?
Sorry, I didn't get it. You want me to speak in Portuguese or in English?
No, in English, because it's just
Okay,
us
okay.
who's
All right.
but yeah, thank you for coming, and thank you for taking time to speak with us here today. We really appreciate it.
Well, thank you for inviting me as well.
So we will start in a couple of minutes. And how are you currently in Lisboa, or where are you currently located?
Yes, I am. Right now, I'm in Lisboa, in Marvila, right next to the river, actually, to right next to the big bridge, to 25th April bridge.
Oh, nice. It's
That's
pretty.
Yeah.
beautiful. I've been there a couple of times.
Sorry, it's my first time that I use Clubhouse. I'm not really familiar with how it works, but uh, I guess it's kind of like a voice chat room, isn't it?
Yes. So, okay. Yeah, exactly. So you found the most important button, which is the
Mike,
unmute button.
The mute
So
button.
we
Yes.
can
All
hear.
right.
Then there is a chat option. So all the way on the bottom on the left, there's like a speech bubble with the number 6. So if you press on
Mhm.
that in the chat room, so there you can chat with everyone, or people that are maybe in the noisy background also, they can comment and ask questions there instead of speak.
Mhm.
Um,
I saw.
and then we have on the top here, I posted the link of um, the
Oh,
Paleomosh.
Paleomars.
Yeah,
Okay.
but
Mhm.
I will switch it
Awesome.
later to the paper when we
Mhm.
start speaking. And
All right.
so how it works, if when people click on everyone, it's not like a screen share. So when people click on the link, everyone has to do it themselves. So if you like refer to an image in the paper or something like that, that's really helpful to
I see.
say, which number, table number or image number you're on. So
Mhm. Okay. Sounds good.
yeah, those are the most important, but there's a lot more, but I don't think, for now you would need it. Or
Okay.
so.
Mhm.
Oh, it's 2. Okay, we can slowly start. And okay, I'm sorry, I was sharing. Okay, welcome everyone to Science Society, and of course, a special welcome to you, Ricardo. And before we start, let me give like a very brief introduction to the audience, and then we'll go from there. So, Ricardo Araújo. He's a paleontologist and the co-founder of the original Niasa project, and is now the principal investigator of the Paleomarsh project. And he did his PhD at the Southern Methodist University in Angola, Plesiosaurus and Sauropterygian functional
Yes,
morphology under
that's right.
Fulbright scholarship. Sorry.
Right.
He's currently a researcher at the University of Lisbon in Portugal, and he led several Palniasa expeditions and supervised, trained a lot of students in Mozambique. And he also designed the paleontology laboratory and monitored its construction and setting up this lab. That's really impressive. So we are very honored having you here. And before we start, usually we ask a couple of interview questions, if that's okay with you.
Yeah, sure. Thanks for the nice introduction, by the way.
Yeah, thank you for being here. And so we usually ask, because we kind of think it's really interesting for people that are maybe not doing research or science, like, what led you to choose science as your path for your life? Like, was it something you always wanted to do? Was it some book, a teacher, some class?
Yeah.
What happened?
Well, it's that's an interesting question. That I've never thought it in with those eyes, because I'm not even sure if it was me choosing science or science choosing me, because I was very young when I decided to be a paleontologist. I was 4 years old, and yeah, I've always wanted to be a paleontologist, and I am basically a kid that never really grew up. I, I still have the same fascination for dinosaurs and extinct uh, animals as I had when I was a little kid, opening up the chips packs, finding my first dinosaurs in there.
That's wonderful. So you went with your passion and curiosity. That's so nice to hear, because actually that's what most people say here. When most speakers say something similar, like that. That they just
Hmm.
follow their curiosity. So it's kind of, we are very lucky to have a
Yeah,
life that we enjoy. And
absolutely.
what led you to the current research, the specific project, or also organizing this Paleomars lab? Like, is there maybe a story? Was it easy to get funding for it? Like, what's the background story there?
Do you want the short version or the long the long story version?
It can be also the long one, because it's kind of unique that we can just have a conversation here. So it can
Okay.
be something in between, long, however you would like.
Okay. Yes. So I've always wanted to be a paleontologist, as I said, and one of the things that paleontologists do is to go to the field and find extinct creatures. When I went for the first time to Mozambique, I was not even starting my PhD, so I wasn't even sure about the route to take in terms of my research, the direction rather. And so I was at the time I went to Mozambique. It was really the first expedition in like 50 years going to that country to find not dinosaurs but mammal ancestors. But in the same year, I was invited to do a research project in Angolan Sauropterygians, those weird named creatures, which are basically marine reptiles from the Mesozoic. but I wanted to be able to develop the paleontology in another country. In Europe, we are pretty lucky in Europe and in the US, because there's already kind of an infrastructure and a critical mass. But in countries like Mozambique, there's no such thing. Really, the research is very little, and and basically there's very few people even considering really doing research, because it's not on top of their priorities. For instance, Mozambique, it's a country that's dealing currently with a lot of war problems in the north. It dealt with a long civil war during many years. There's starvation, there's a lot of economical problems in that country. So it's research, it's really not one of the top priorities. But the way I think about research, and especially paleontology, in a country like that where the reality is very different, is it can be a catalyst and a booster for economical growth. For instance, there's places in France that attract millions of tourists every year, and they literally make certain areas in France, and even in Portugal, or any European countries or North America, that bring a lot of people. And what I saw with that is that it's a tourism promoter. So the way I like to think about the PaleoMos project, and instilling research by Mozambicans in Mozambique, is kind of like a way to attract tourism as well, because when we find new things, especially stuff like mammal ancestors from like 250 million years ago, it also sparks the curiosity of people, of Mozambicans themselves, first of all. So they need to be firstly the ones interested on those matters, and that's the reason why focusing on the education is so important. And then later for bringing tourism as well. So it's a really long shot, I know, but that's kind of more or less the idea. So, but to go, I mean, I'm jumping back and forth, but I guess that's how it really is. And then when I went first to Mozambique, actually, it was really a first step we needed to find something, basically. So in 2009, I was 23 years old at the time, and we just needed to find something, basically, to justify future expeditions, and to justify that Mozambique was actually worth paleontologically speaking. There was enough fossils and enough material that would justify developing things further. And after like 3 weeks in the fields, and without finding nothing, really, I was in my last week until we found the first fossils. It was really frustrating and complicated, because I was in a really remote area in Nyassa. Nyassa is the poorest province in Mozambique. And there's really literally nothing. It's just small villages with very few people living there, just doing like very traditional. It's not hunter gathering. It's like, but it's close to that. They are just doing some farming, some little farming, and trading goods. Often money is not even involved. There was no electricity. There was literally nothing. And I was hiring local people to just guide me through more or less the field, although I knew more or less the areas to go. And they were just not believing in me. They were like, this guy's just insane. This guy's just crazy. He's coming here looking for bones of animals that are like 200 million years ago. That's completely insane. This guy is crazy. So I was like, week after week, their disbelief in me just grew ever more, and even I started to doubt myself. But the I knew there was material there. I knew there were fossils there because there had been reports of those fossils in the initial geological reports from the area. So I knew there was something. It was just that I was not being able to find those locations. But lucky or not, towards the end of the expedition, I was again just completely alone and disbelieving myself, and everyone around me was just not believing either. I just flipped the rock, and there it was, like a complete, beautiful skull of what turned out to be a new genus and species, which now we call the Nyasodon fumukazi. So the queen of Nyasa, basically. That's the name of the fossil, if you translate it to the local language. And it's that name because in that particular region, the tribes that live there, they are matrilinear. So it's actually the women that kind of uh, operates things more or less over there, or it's passed through the female lineage. So it's quite interesting. So we decided to call it the Queen of Nyasa because we don't really know the gender of that fossil. So with that, we had like the first good justification to actually go back to the fossil sites and to apply for funding, because that's really what we need to like conduct these field campaigns and these field expeditions. And so we initially applied for National Geographic in what was called at the time the Young Explorers grants. I don't think there's such grants anymore. And we applied to those. It was only $5,000, but it was enough to go back again next year. And sure enough, we went there again, and we find more stuff in different locations. So and it kept like we kept going, and we kept being able to justify more field expeditions, and also with fundings from we managed to work very directly with the National Museum of Geology in Maputo, the capital of Mozambique, and they also brought in funding. We also applied for funding via Mozambique to start developing the students there, to send them to different places to get some more training. And it kind of like really grew organically, and we started with like $5,000 to go to with the National Geographic grant, and a couple of years ago we got Aga Khan Foundation grant, which was 300,000 euros. It's not yet a lot. There's still those multi-million multi-million funds to apply for, but it's been enough to like justify to the local authorities to build up a new lab, to buy equipment, to furnish the whole structure in Maputo, to get training for many different Mozambican students. We are lucky now to have like the first two Mozambican students that are doing their PhDs in South Africa at Wits University. So it's really, I mean, yeah, I'm really, really happy to see that what started off as just like a mere hope to find something, it's now really materializing into making paleontology and science an interesting and valuable thing for the Mozambican people. So yeah, that's more or less what happens.
Wow, so impressive. Like you should be. That is really so impressive, and I really congratulate you for going through with everything. It must have been, a lot of adventures and struggles in there,
Mm-hmm.
and that you kept.
Mostly struggles, to be honest,
Yes,
because it's very it's such a different reality in Mozambique, and it's almost like we operate in different wavelengths. When I arrive to Mozambique to do anything, it's literally I have to tune in to the wavelength, and they have to kind of tune into to mine too, because that's how any relationship really works. And but yeah, it's been really a lot of struggling and a lot of emotions, but yeah, it's slowly getting there. I mean, we started in 2009, so it's been over a decade since we are doing this, so we better have some results as well.
well, you do. This is a really impressive publication that I posted it up on the room, and it's a major, I think, result that you managed to get there. So you're really putting Mozambique research in
Hmm.
a major scientific paper and on the map. I think that I didn't. I'm not sure.
That's
I didn't.
precisely the idea.
Yeah, that's huge. Like it's a huge achievement. Congratulations. So
Thank you
happy
so much.
for you. So
Thank you.
yeah, feel free to talk here, tell the article, about the research, like what the findings are, and it's up to you if you welcome questions in between, or if you want people to wait a little bit until you can like bring your message over, and then we'll open up for questions.
Yeah, people can just interrupt me. I think it's just more dynamic. I have no problem with that at all.
Great, thank you.
But yeah, if anyone has a question right now, just feel free to interrupt. I have no issues with that.
I have a very important question.
All right.
What's the tastiest food out in Mozambique?
The tastiest food, I would say it's like fish directly from the sea.
There you go.
But I mean, that's really the best food you can have, because they have awesome seafoods, and lobsters and big shrimps and all of that. But unfortunately, most of our field areas are like really, really inland. The most inland in Mozambique that it's possible. So we rarely have a taste of the good stuff. Only one year we had part of the expedition was in the coast, because we found like a new lignite deposit. Lignites are kind of like the early stages of coal, basically. And yeah, we did find this location, and lucky enough it was just right off the coast. So yeah, on that time we did try some nice seafood. But yeah, if you go to Mozambique, you gotta go to the fish market, and you gotta eat all of that good seafood.
Awesome.
Okay, going now to the paper, and how come Mozambique is somewhat involved? Of course, Mozambique is not the main focus of the paper, but it happens that Mozambique contributes with fossils to some of the results of the paper, and that's why Mozambique kind of comes into the picture. So basically, what we found was when do mammal ancestors, we find out that it's not mammals that initially were warm-blooded. It was their mammal ancestors, their mammalian ancestors, that were warm-blooded. So being warm-blooded is basically you're able to produce your own metabolism uh, at, at a very constant rate and a very high rate as well. So the rates of metabolism for endotherms, we call endotherms any warm-blooded animal basically. It's way higher. It's many times higher than an ectotherm. Ectotherms are like the animals like lizards or snakes or turtles that kind of mostly depend of the environmental temperature for them to do anything really. Fishes are also ectotherms. So the only groups of animals, major groups of animals that are endotherms, so they warm-blooded, are mammals and birds. So we wanted to see when did mammals first evolved endothermy. So when did they first evolve this warm-bloodedness? And we found out it was actually some 30 million years before the origin of mammals themselves. So it was their mammal ancestors that had this. So the big question is, how can you really measure body temperature or estimate body temperature in an extinct animal? Because we cannot just simply stick a thermometer in a fossil, because that's just going to tell us the temperature of the rock, obviously. So we had to use like a very unconventional, I would say, proxy, which is the inner ear. So our ear is composed of two main things. The ear is not only capable of hearing things, that's one part of the ear. The other part of the ear works as kind of like the sense balance. It's called the vestibular organ or bony labyrinth, or bony labyrinth. And the other part is the cochlea, which is kind of like a coiled structure, and that's for hearing. So your inner ear has these two major functions, and we just focused in one of the functions, the vestibular organ, these semicircular canals, which so the vestibular organ is composed of these semicircular canals. And these semicircular canals are called semicircular because they are kind of circular most of the times, and they are orthogonal to each other. And the fact that they are orthogonal to each other, still backing up a little bit. So these semicircular canals are filled with a liquid called endolymph. And when you rotate your head, this liquid inside of these canals is going to kind of slosh around. It's going to move in the opposite direction that the head is moving. And the canals are kind of orthogonal, so you have we humans and animals operate in 3 dimensions, so they kind of detect each orthogonal canal detects the accelerations of the head into the 3 dimensions. That's basically how it works. So there's this liquid called endolymph, and the equations that govern the sensitivity and basically the biomechanics of the inner ear of the semicircular canals is dependent on temperature, because the viscosity of the endolymph is dependent on temperature. If you think about it, any liquid is dependent on temperature, of course. If you start let's imagine if you put some oil on a pan, and at room temperature is very viscous, but when you start heating it up, it starts becoming less viscous. It starts to be runnier, basically. And that's really the principle. So when temperature increases, such as the body temperature of mammal ancestors increased, that had an impact on the viscosity of the endolymph, on the runniness of the endolymph, because it was operating at a higher temperature. It's just really a pure physical constraint. So what needed to happen for the function of for the biomechanics of the semicircular canal to be maintained was to change the morphology or the geometry or the geometry, basically the size and the morphology, the size and the shape of the inner ears needed to change to compensate for the increase of body temperature. So that's the theoretical principle. Of course, we had these ideas before running the experiments, and it turned out that it works. We were actually kind of surprised that it really the temperature could be estimated on the basis of the shape of the semicircular canals. And we know it works because we tested in like several hundreds of extant animals, so animals living today. So we measured the biomechanically relevant uh, um, parts of the inner ear. We measured all of them for 300 something animals for the three canals and several times. So that's like several thousands of measurements. And by looking at we know at what body temperature animals operate today. So typically warm-blooded animals, they are called warm-blooded for a good reason, because they do have body temperature that is higher than for the cold-blooded animals, like lizards and so on. So their body temperature is significantly higher than for those of ectotherms. Although there's exceptions, there's many ectotherms that actually like to operate at very warm temperatures, but those are more the exception than the rule. If you look at one of the pictures of our paper, it's actually figure 3. There's these two. If I think you all have access to the paper, right?
Yes. Mm-hmm. Yeah.
Okay.
It's
So
pinned up.
yeah, so in figure 3, in one of those uh, probability density plots, where you have basically two. It's not two histograms because they are really two curves. You can see that the blue ones are the cold-blooded group, and the red ones are the warm-blooded group, and they separate really, really well. So we managed to test empirically that this proxy actually works, and it works with an astonishing precision of some 90 something percent for groups. When I say for groups, is for groups of animals, so clades of animals, because this metric, what we call the thermal motility index, works really well for groups and not so well for species, because species for species level, the signal from locomotion, because basically when you're looking at the inner ear, the inner ear is a system to basically measure the rotation of the head. So locomotion has a big impact on the functioning of the inner ear, obviously. But we, by working on the equations and by basically pulling apart the information that we need, it works really, really well for the temperature. But for species, the locomotion effects kind of like overrides the signal, and it doesn't work as well, but it works still pretty well. But for groups, we can have some 90% accuracy to detect the to detect basically, if you give me a group of animals and the inner ear of a group of animals, and we make the measurements, we can basically tell you with 90% accuracy if it's going to be an endotherm or an ectotherm. And this is done by then looking at the logistic regression, but that's kind of like technical detail. I don't think it matters so much. So yeah, it works pretty well for extant animals, and then of course we want to test for extinct animals. When you look at extinct animals, you have several different complications, because oftentimes the inner ear is not entirely preserved. Sometimes it's deformed. Sometimes it's not preserved at all. Many scans. Actually, we have a section in the paper, in the supplements, saying how many unsuccessful scans we've done. There's many scans that basically did not work. We put them in the machine. We try to scan them, and then either there was no contrast or that region was not preserved. So it was really like an iterative process of like going back and forth, of refining the model and increasing the sampling, and this over and over 5 years that it took to complete this study. So we started to amass the data for the fossils, and we ended up with some 56 or 60 something. I can't recall the number. It's around 50 or 60 specimens of fossil synapsids. So synapsids it's basically the lineage that leads towards mammals. So it contains mammals, and it excludes basically reptiles, all the reptiles, including birds, because birds are reptiles, because dinosaurs are in that lineage, and dinosaurs are relatives to birds. So so we looked at these fossils, and we kind of started to trace back when would this origination of endothermy would happen in the evolutionary tree of synapsids, which is the lineage that includes mammals. So we found out that it was in this clade called Mammaliamorpha, which in technical terms is Tritylodontids plus Mammalia. It's this clade. Tritylodontids are, if you would look at the skull of a Tritylodontid, it is kind of already hard to differentiate it, unless you are really like a specialist. It would be hard for anyone to really see the difference. But these mammaliamorphs have an origination dates much earlier than the actual clade of mammals. When I say clade, is like the group. It's the monophyletic group of mammals. So that's basically all the descendants of mammals. So it's in this clade, and we found out that the acquisition of endothermy was actually very sudden in geological terms. It was in about 1 million years, and we tested this several different ways. We actually purposefully used gradual models to see if they would retrieve better results or not, and it was very consistent that the best evolutionary models that would explain the origination of endothermy, so of warm-bloodedness, would be like a very fast acquisition. That was one of the surprising aspects of the paper, and yeah, like in a nutshell, that's more or less what we did.
That's really impressive.
So,
That is so accurate, and that you can
mhm.
distinguish so well.
Yeah,
That's
indeed.
really interesting. And so basically, do you so what happened to basically the timeline? Did that change, like your inside the timeline of when we think that warm-bloodedness started developing, or is that, do you still need to find more, bones?
Uh, You mean what do you think triggered the such a sudden origination of endothermy?
Yeah, exactly. Like, did something happen
Okay,
or yeah.
we have, well, that, again, we are working with fossils and can only work with their bones mostly, unless you have like very exceptional findings. Sometimes that does happen, and you can find fossils with hairs and even like really soft tissues, like guts and like brain parts and but that's really rare. That's much more the exception than the rule. And so what we believe it happened was during this periods of probably 1 million years was the origination of fur, but fur in the sense of pelage as an insulatory device, let's say, uh, because if you are producing your inner heat, you need to preserve it, and fur and feathers are really some of the highest insulatory the materials of highest insulatory properties. We all know that there's we use wool as like winter clothes, so that or even sleeping bags that are made of feathers, so they really have amazing insulatory properties. So and that's one of the things, and the other thing is the sarcolipin mediated thermogenesis, which is basically a metabolic pathway that allows cells and mitochondria to generate their own heat at a constant rate. So we believe that these two kind of key innovations had to occur at the same time, because if you're producing your heat and you're letting it dissipate, it kind of doesn't make any sense, because the energy for any animal is obviously very costly. And of course, I didn't mention this, but the biggest difference between ectotherms and endotherms, so between warm-blooded and cold-blooded animals, is that the warm-blooded animals have a big disadvantage. It's because energy is very costly, and you need to forage for more foods and to have a higher intake of foods, comparatively speaking to ectotherms. Ectotherms, they very often brumate, which is another fancy word for hibernate. They are not active through most of the year. We in most places except like in tropical regions, but like in temperate regions like in Portugal and Europe and North America, lizards, for example, are mostly seen during the summer times, because they simply cannot heat themselves enough during the winter to actually do any activity. So they enter some kind of state of hibernation. Of course, being endotherm allows you to explore your niches throughout the year, and kind of like be more independent of the winds of the climate, basically. Where was I going with this?
Yeah, I think you explained like what the circumstances were to kind of support this
Yeah,
future.
exactly. Yeah,
Yeah.
yeah. So basically, two things had to happen physiologically wise for mammal ancestors to become endotherms. We simply detect that manifestation by looking at the inner ear of these mammal ancestors, in how the inner ear size and shape change throughout time, basically.
Yeah, thank you for your answer. And yeah, want to invite other people to ask questions. Please flash your microphone if you have a question. Yeah, please go ahead.
Hi. First of all, thanks for taking time to share your work with us, and your backstory
Hmm.
was super interesting. Um,
Thank you.
so I've been working in the auditory system for a while. I currently work in the cortex, but I spent years doing biophysics and work in in the cochlea mostly. So this is super interesting, and I really like the arguments you were making about the temperature and the effects on microfluidics. I'm curious because I was really locked in on modeling the micromechanics of cochlear hair bundles and comparing them across vestibular hair bundles. I'm curious if you've thought about whether there's any parallel with the change in the shape of these canals with changes in hair bundle morphology.
Well, I would. That's a hard question because we cannot really go to the micromechanics. We have to stay at the macromechanics. So at the micromechanics, we are basically operating in terms of the upper corner frequency of the semicircular canals and the sensitivity. And what to all right, to answer straight to your question is we don't know. We really don't know, and I'm not sure if we can know because we would need to have hair bundles preserved in the fossils. So what we can look is at the size and dimensions and morphology of the inner ear of the semicircular canals. What I can tell you that we can deduce is by looking at the membranous labyrinth and there's in the equations there's some variables like the cupula stiffness, but that's still not micromechanics. It's still at the level of macromechanics. So there's it is possible to estimate those things, but it's I would say it's impossible to really ascertain that. But my prediction is that there wouldn't be much changes at the micromechanical level. I'm saying that because we don't see much changes in the micromechanical level between species, extant species. Because, for instance, for the hair bundle cells, the system that it's used is like the toadfish, so it's like a very basal vertebrate compared to a human, and that's the model for humans. So in terms of the micromechanics, I don't think those changes would my suspicion is that those changes wouldn't be too significant, but it will be impossible to answer that directly.
Yeah, I wasn't really trying to step too. It depends on what you mean by micromechanics, I guess. I wasn't really trying to get at that. I do have some currently unpublished work that gets at that. The only thing I would push back on a little bit is there are significant micromechanical changes even within a single vestibular organ across, spatially across the organ and certainly across the tonotopy and the cochlea. And there's big micromechanical
in the
differences.
cochlea, but we are working in the vestibular system.
Yeah, there's a big difference between vestibular and cochlear micromechanics, and then there's big differences across species. But I was more wondering if there was like a corresponding systematic change in like gross hair bundle morphology, like size and shape of hair bundles corresponding with changes with shapes in the canals. But yeah,
the ideal person to answer to this would actually be Romain, who is unfortunately not here because he's more like the semicircular canal biomechanics expert. I know this, but not at the level of depth that goes down to the level of micromechanics. what I can tell you is really all the variables that deal in some way or another with micromechanics in the equations that govern semicircular canal biomechanics, so like upper corner frequency and sensitivity, do not can be estimated from the membranous labyrinth. Not the so they can be estimated towards they can be measured in the membranous labyrinth and be estimated for the bony labyrinth. Um, but I'm not sure if they would have much of too much of a big difference. I can ask my colleague or we can stay in contact and we'll try to explore that a little bit if you want.
Yeah, sounds great. Thanks for answering my question. And I'm going to share your work with my collaborators. And this is
All
very
right.
interesting. Thank you.
Mm-hmm. Thank you.
Yeah, thank you. Dr. Shah, Joyce, Frank, did you have a question?
So if nobody else has a question, I have now one.
I
Do you have
can ask one
Namanu?
question. Yeah, yeah,
Yeah,
yeah.
yeah. Go
So
ahead.
maybe it's I'm not sure it's within topic, but I was reading something about chameleon and something like this animal. I mean, it's a bit puzzle of like how these animals change color. What is the evolutionary? What is the evolutionary perspective from for this particular animal to get this color? In one of the paper I read, it's because it's trying to change the temperature of the body, because when you this dark color you can have more temperature. At the same time, if it's light color, you can radiate more temperature. So I was asking if you can give a hint about like how the evolutionary trait actually evolved, and I mean how it got the ability to change the colors and also the temperature.
Uh, yes. All right. So a chameleon can change colors and maybe heat up using basking and using darker colors to attract more heat, therefore to increase their temperature for being active. So yes, indeed, a chameleon is like your typical ectotherm that is basically actively thermoregulating by basking and doing all these behavioral things to heat up their temperatures until it reaches uh, their preferred temperature. So that then they can go and do whatever they need to do. Eat or forage or whatever. So, so your question is more like why would they have the body temperature that they have? Or,
Uh, I mean, my question was like, is there any environmental feedback? Like, if you say, okay, evolutionary, like Darwinian point of evolution, like random mutation, but how is it possible to change the color or according to the environment or the
I
temperature?
mean, they might change the color for regulating the temperature to get to their preferred temperature, right?
Yeah.
Uh, but it's still that's like still a typically ectotherm like behavior uh, because again, like typically ectotherms, they have a preferred temperature that they like to operate, so they need to reach up that temperature so that they can then be active, while endotherms, they have a constant inner metabolic system that raises the temperature and maintains the temperature constant. So I'm not sure if your question is more like, okay, if an ectotherm changes temperature and it still can operate within a certain range, how does that work? And one thing is the plastic, the plasticity of the physiological components of the inner ear. They can operate at a range of temperatures, but they have still a preferred body temperature that they like to operate the best. That they operate the best, actually, and that's been demonstrated by several papers, actually, in terms of like locomotor performance. Of course, there's like uh, it's really like a curve with a maximum. If you keep raising the temperature, a lizard will keep improving the maximum speed that they can go, and then it starts to decline. And it works more or less the same way for for the inner ear. It has it's like a curve. It should increase their performance until a certain temperature, and then if it starts operating at too higher temperatures, it's going to decline the performance, and that can be demonstrated by looking at the equations themselves. But one thing is this plasticity of the physiology. Another thing is the different time scale that we are talking about, and the time scale that we are talking about is evolutionary time scale in the order of millions of years. So we are not talking about little physiological changes to the inner ear and how like say in terms of micromechanics, things can change, but really at the large evolutionary scale, how did the inner ear change in terms of their gross morphology and dimensions? And what we do see in the fossil records, if we trace back the evolutionary history of these mammal ancestors, is that the size of the semicircular canals decreases, and that's predicted by the governing equations of the biomechanics when the temperature increases, when the body temperature increases. I'm not sure I was able to. I think this was more or less what you wanted to know,
Thank
right?
you. Thanks a lot. So
Okay.
it was wonderful, Tom. Thank you.
All right.
Yeah, Frank, do you want to go ahead now? Thank you.
Well, I think Dr. Shah, you opened your mic. I think maybe you want to ask a question first.
Yeah, you can go first, or I can ask. Okay, thank you so much, Ricardo. That was a really fascinating work. And my question from you is about the optimization, because we are talking about the inner ear system, and I'm just assuming there is a optimization needed there, especially when you are talking about the temperature. And I was wondering, do you have any information about the electromotility and the distribution of the protein in regard to your research or not?
I mean, again, with fossils, we are limited in terms of the amount of information we can have. Of course, there's this potassium channels at the very micro. I think that's even nanomechanics at a nanomechanical level. That need to be regulated for the hair bundle cells to work, but that's at a very different level than we are operating. We are operating at a macro mechanical level because that's really the only types of information that we can extract from fossils. So we have to bear in mind that we only have access to information of the bony labyrinth. It's not even the membranous labyrinth, because the endolymph runs within the membranous labyrinth. And we don't have access to any information such as the cupula, the hair bundle cells, those potassium channels, and all of that. So even for extant animals, that type of information is really rare. Even more so for to go down at those levels at really chemical changes. As far as I'm aware, I mean, maybe there's new current research, but I think, yeah, I think that's about the best we can do really at the moment.
I'd say one quick thing. As you said, even in existing animals,
Yeah.
definitely in humans, we do not know the identity of the mechanotransduction channel. We don't know what those
Yeah.
proteins are. There are a couple labs in Harvard that are kind of making the Kool-Aid on a current theory. There's two proteins called TMC1 and TMC2 that are
Hmm.
hypothesized to be part of that channel, but
Hmm.
it's under heavy debate. So we know some pieces of the like tip link mechano sensitive channel assembly, but we really don't have a full picture of what's going
Yeah.
on. Yeah.
Yeah. No, but that's going down to a level that we are not really operating. We are operating at the largest scale possible, and it's still for an entire dimetrodon, which is a 4 or 5 meters long animal. We are operating at millimeter scale. Uh, for an animal that is 300 million years old. So we really have to operate at a macro mechanical level. We cannot even think. I mean, we can deduce things and we can extrapolate how they might work, but we cannot have access to any of that information at that level. The thing is, in the end of the day, what's really surprising and what we really enjoyed is the fact that by looking at the macro mechanical level, still there is empirical strong evidence that the thermomotility index, this matrix that we are using, works with 90% accuracy. So that's something that needs to be taken into account. So temperature does have a huge impact in the macro mechanics. So if it has an impact on the macro mechanics, it will certainly have at the nanomechanics level and certainly have at the micromechanics level too. So yeah, I mean, uh, yeah, it's we have to work with what we have, and what we have is fossils. And even to get information from those fossils, I tell you, it took us a long time. Took us many years to assemble this data set. So, I mean, it is what it is. I would love to look at proteins in these fossils, but I mean, we can't. That's the reality of things, unfortunately.
Like, if I would design it, right? I'm not a audio circuitry expert and never was, but if I would design it with having more liquid faster, I would just add more inhibition to the system, no? Like some more inhibitory neurons and receptors. Wouldn't that
From
wouldn't
a
that be enough?
from a neurological standpoint,
Yeah,
well,
that would probably.
it seems that animals did not opt via that way. I mean, if the morphology of the inner ear really does change,
Oh no,
that seems that it was effectively the
yeah,
option that animals took.
yeah, that. But then also, don't you? Would you think that there's still like just more signals coming in still, like, and then
But
you wouldn't
clearly,
maybe
the
add.
temperature signal seems to have an overriding effect on this, for sure, because otherwise we wouldn't have such a clear separation between ectotherms and endotherms,
Yeah, yeah. No,
right?
I don't mean like just like the anatomy, but additionally, maybe you would add to the neurological circuitry some more inhibition to the system. I
Mm-hmm.
don't.
Yeah, yeah, yeah.
Maybe it doesn't make sense, but
Mm. Uh,
wisdom. Do you think that
Uh,
makes sense?
what I would say is I'm very biased. I think the hearing organs are fantastic. They are as finely tuned and optimized uh, of an organ as we know of. Like the auditory system, for example, is tuned to up to 9 octaves, which is in contrast to less than an octave in the visual system. It spans multiple orders of magnitude, and it's very, very sensitive. Cochlear hair cells can detect vibrations of the tympanic membrane that are less than the width of a hydrogen atom. So things have been really, really, really finely tuned. It's broad dynamic range, high sensitivity. This is the kind of thing engineers just dream of designing. So in terms of the front end, I don't know that there's much more to be optimized. But certainly downstream, there's
But
opportunities for
now
neural
that
integration.
reminded me of one thing. I'm not so sure about the neurological aspects of it, but one thing that did change, and that birds, for instance, had taken a different evolutionary route, was instead of changing the morphology of the morphology and the size of the semicircular canals, birds did change the viscosity in terms of the chemistry. Literally, the chemistry of birds, um, uh, bird endolymph is different from all the other animals. So, and birds are indeed endotherms, and they took an entirely different route, evolutionary route, to become endotherms, or to readapt their inner ear system to the new reality of warmer temperature.
The
But
other big
I mean,
the
this is
other big
again not talking at a neurological level. This is really talking about just the inner workings of the semicircular canal system.
another
So I got a quick question about.
I'll just make one last
Sorry,
comment.
go ahead, Wissam.
The other thing that's of interest, a difference across mammals and birds, is birds can regenerate hair cells. So everything I was
Mm.
saying about, the precise optimization of the mammalian system, that has to work with the constraint of not being able to regenerate hair cells. So it
Mm.
has to do all of that and be
But
robust
that's for the
to.
cochlear. That's for the cochlear system, right?
Yes, same for vestibular.
It's
The
okay.
labs that are going after how to regenerate hair cells in mammals are studying, vestibular end organs
Okay.
uh,
Okay.
of birds. Yeah.
Yeah,
This would
usually when you have a more fine-tuned system that is more precise, you basically have more variety of inhibition. Like you have more different types of interneurons and also more subtypes of GABA A receptors and so on. So that's why I thought that there's probably a heavy complex inhibitory system than the normal
So,
person.
yeah, now that you say that again, Katerina, it does remind me too. This is stepping away from vestibular, but cochlear. Sorry, that's what I know best. There's actually
Mm-hmm.
two types: inner hair cells and outers. Inners send signals to the brain, and the outers amplify signals, and they receive feedback from the brain. So there's a mechanism
Mm-hmm.
for
It's a
the
feedback loop.
dampening of sensitivity along the cochlea from the brain. So there is a sense of this top down inhibition that you're talking about, Katerina.
Cool. Thank you. Maya, go ahead.
Speaking, it'd be interesting as far as the viscosity changes in the birds. You say, okay, they changed it. So one question would be, why did they change it? Is it because they're gonna, basically go up in elevation and down and adjustments there? That's one. And then the second one is, what about those diving birds, right? You know, those birds that could dive down into the water and hunt for fish, but they go down pretty darn deep, right?
You know, the types of questions that I'm getting, they are. I mean, you know how many viscosity measurements have ever been done for the endolymph of birds? They have only been done for one species, which is the pigeon, and for 8 samples. Okay, so the amount of research on this stuff, it's really really limited. I mean, I have no idea if diving birds. My suspicion would be that, overall birds would have more or less the same viscosity, because they operate all more or less at the same body temperature, and they do more or less the same kinds of things. Of course, this is like a suspicion and a guesstimate, an educated guess. Really, that's really what it is. But it's no more than that, because there's really little. I mean, as I said, there's only one paper ever published on the viscosity of the aniline for birds. So I mean, and that's actually one of the things that our paper exposes is that there's very little stuff done across entire groups of animals in this type of research. I mean, we are paleontologists and we are working with our systems and what we can do. And actually, we did try to talk. Maybe some of you know Richard Rabbits, who's from the University of Utah, and he's kind of like a little bit of the Pope in terms of semicircular canal biomechanics. And we did try to work with him in this respects. Like, let's try to do some measurements of viscosity and so on. But in the end, he had other research priorities, and it did not work. But there's one of the things that we found out is that there's a worlds of information to be known. Really, that was something that our paper really shows. Of course, it's not to the eyes of say someone that never did research on this and that reads the paper. But that's really one of the things that we highlight. There's a world of information to be gathered, basic information like uh, even membranous labyrinth dimensions for many animals. It's not known. We actually presented the first measurements of the membranous labyrinth for, or not the first, but some of the first. There were like 3 or 4 previously published, and then there's the Jones and Spells paper, which we also used. But for many measurements in 3D, we were actually the first ones to produce an entire data set from the membranous labyrinth for 35 species or so. So I mean, we really, I think that we made really important strides in terms of like comparative. Physiology, if you want, of the in a year for a lot of different extant animals, and really how we deduce the bony membranous relationships were, this is like entirely new stuff. These relationships were for the first time published, were published for the first time. So yeah, I mean, we've done what we could to have like a firm statement about like bony membranous relationships, for example. But we are well aware that there's a world. I mean, this is an entire many careers over that can be dedicated to like explore the membranous labyrinth, the cupula stiffness for every single animal, the viscosity of different groups. So far, for the entire animal kingdom, there's only 8 species known for their viscosity. 7 of them have water-like viscosity, including humans. And then there's birds. Birds clearly stand out. Birds, when I say birds, it's like the pigeon species. Okay, it's Columbia livia, and that's all. But I mean, yeah. We did try to tackle that before to have even more assertive conclusions about about this. But first of all, we were not interested in birds. we were interested in mammals, and for mammals there were like 3-4 species published, and all those 3-4 species had water-like viscosity. So that's what we can do.
Oh yeah, we totally agree that you did, really amazing, very impactful research. We just are contemplating about um, what else, what's next. So I really appreciate you summarizing, the amount of work and impact and so on that you did with this work, and it's for sure a lot of work and hard work. So we really appreciate that for sure. I know Frank had a question. Do you want to go ahead, Frank?
I have kind of two questions, but it seems like with a question that Maher stated, I think I'll get a similar question because I'm also going that direction. So I mean, right now I'm more of a social scientist, but I trained as a sound engineer, so I'm very
Okay.
into the speed of sound thing, and and you touched the motor coordination, navigation, spatial awareness. These terms are in the paper. So I was also wondering if there's the speed of sound in air is 343 meters per second, and in water it's 1,480. So I was wondering what the temperature of the blood had as an effect on this navigation system. So that was kind of my first question, but I guess the answer would be kind of in the direction that Maher got.
But the thing is, the temperature of the endolymph is the temperature of the body, and not the temperature of the medium that the animal is immersed in, whether it's air or water or I don't know, lava. Whatever. The body temperature is really what matters. It's not so much of the temperature of the medium. Unless you are asking more things along the lines of the cochlear system, which is the auditory system, and that would not be a question for me. So that
I
would be
see.
for the cochlear researcher that was talking before.
Yeah. With and I we are having these discussions, so I'm always interested into that. So, but as a paleontologist, I hope
Yes.
I get that correct, right? So, just a more basic question. I'm also interested into when the tactile sense split from the auditory sense. So maybe that is way outside of the scope of the paper. So I'm just interested in your take on that. How did that happen? Because I have a lot of discussions on that. That the touch and hearing, that there are similar cells
Yeah.
involved, and so if that, how is that similar or are different in your understanding in the evolutionary sense?
Well, that's one of the most marvelous stories that paleontology can tell, and unfortunately, I'm not a specialist on this. But what you can see very, very clearly in the fossil records is the evolution of the middle ear in the fossils. You can actually see the migration of the mandibular ears, mandibular bones, migrating in to form eventually the middle ear. So those three bones that compose the middle ear. And basically, yeah, it was what paleontologists can tell is kind of like the timing of that occurrence and how did it change. Eventually, I'm not so much aware if there's people really working on the biophysics of how that sound transmission stuff works. But what happens physically speaking, and what I can tell you because I just know a little bit about it. I'm not a specialist on this area, but yeah, the mandibular bones, so the jaw bones, the posterior jaw bones, so the back parts of the jaw bones. So yeah, all right. If you look at the reptile jaw, it's composed of many different bones. Okay, there's like the angular, there's the cerangular, there's the articular, there's the pre-articular, there's the coronoids, there's a bunch of different bones. There's the dental, where the dentary has all the teeth. So like there's different bones that compose the jaw of say a lizard, your general reptile. But if you look at the jaw of a mammal, it's only composed of one bone. So you have these two extremes. You have a lot of bones in like the ancestral condition, and then you have only one bone in the mammalian condition. And what really happened was that the back part of the jaw, what people think is that and actually you see many lizards doing this today. They kind of put their head on the ground to kind of like here it's not really hearing, because it's more like to feel the transmission of vibrations through the ground. So they put their head on the ground, and when you put your head in the ground, if you are a lizard, the bones that touch are the mandibular bones, so the bones of the jaw. So the posterior parts of these bones started to shorten and shorten ever more, until they become what they are today. Oh my god! And it's really just three tiny little bones. Hello?
So, I guess
I think Ricardo
did
got a phone call. I think he's still speaking, but there's maybe another phone ringing.
Yeah, I am, but there's so much noise right now. I think they're kicking me out.
Mm.
Hello?
Yeah,
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 we're here. Are
we
you
hear you.
okay? Is
Thank you.
everything okay?
Yeah, just 1 second. Hello?
Yeah.
We're here.
Yeah, you're still on. No
Okay,
problem.
sorry.
Do you?
Sorry, they just started music out loud here, and I needed to migrate somewhere else.
Yeah. Well, how much time do you have? I know we're going over an hour now, so yeah. Let me know.
Until all your questions are satisfied. So
Oh wow.
That could be like 12 hours, dude. Just so
Yeah.
you know.
Don't say that. Don't
Like we
say
talk a
that.
lot on here.
well
We got a lot of
then
freaks.
But we also listen.
I don't know, a little bit more, because it's night here already. It's 8 p.m.
Yeah, I'm 1 hour
Yeah.
ahead here in Germany. So
Okay.
As long as you're having fun and you want to keep going, the clubhouse is kind of what we do. I guess we're shifting into more of a social
Okay.
experience there.
Yeah, we can just keep going for a little bit, and then actually I have a dinner, but it's not too formal. So yeah.
So, I mean, one of the reasons I was asking about the birds earlier, I was wondering, like, because when the birds, they dive, they go down, and there's, especially those deep diving birds, right? There's a lot of pressure that they're applying to the ear, right? And
Mm.
especially I was wondering viscosity levels of, having, like, especially with nitrogen going into the, in the bubbles and stuff like that, right? So I was wondering, just in general, is pressure, like how with mammals, are there mammals that in your studies that put themselves in pressure situations, or pressurizing environments, right? Does that affect number one? And then the second thing actually was, as far I learned recently that I guess the nose is more listens to the molecules than it does really smell with the lock and key thing. It's more of a vibration deal. And so, have you noticed correlations between scent and your studies? Right, like are there, like whenever there's changes, are they also changing in the scent as well as the ear, or are those like kind of independent
Okay.
in adjustment?
What paleontologists can know is quite limited in terms of especially the sense of smell, because okay, what can we tell? We can tell the size of the olfactory bulbs by looking at the brain endocast of different creatures. When they are well preserved, they are not too deformed. and we simply have scans that work. I mean, there's really literally some really simple technical issues that sometimes can screw up everything. Um, so that's one of the things, and the other things is like osteological correlates. That's the fancy word to say signals that the bone can give for stuff like whiskers, for example. And whiskers, well, it's not really smelling, it's more like sensing in a way, and it's not necessarily related to the olfactory. Yeah, it's not necessarily related to olfactory bulb, because it's the trigeminal nerve that innervates that stuff. So it's, yeah, that's more mechanical. So yeah, that so to really look at the sense of smell, you need to look at the size of the olfactory bulbs in endocasts, because we don't really have the brain preserved in mammals, in mammal ancestors, in this case. And what seems that happened was that in these mammal ancestors around 200 and so Morganucodon is 230 million years ago. No, Morganucodon is more recent. It's like 200 million years ago. Yeah. They had a big expansion of the olfactory bulbs. Okay. So it seems that mammal ancestors were more like driven by smell than actually by vision, because the optic lobes were not so large. So yeah, what we can say is that around 200 million years ago, there seemed to have been a trade-off, or not so much a trade-off, but an expansion of the olfactory capabilities of mammal ancestors. But you see, that's a kind of like a limited. Maybe this is not really answering or satisfying your curiosity per se. That's really all we can say. I think that's really what the legitimate paleontological answer is. That all we can say is that there seems to have been some kind of increase in the olfactory capabilities in mammal ancestors 200 million years ago. That's all we can say. If you assume, by the way, if you assume that the amount of neural tissue is a good estimate, is a good correlation with the performance is supposed to be taking.
Hey, quick question. Hey, Cecilia, Katerina, I don't remember if it was in this room or if it was in a quantum room. When did we have the room in here with the guy that was doing the microbiome and the AI in the microbiome? Was that here or another room?
Yeah, it was here, but and
Yeah.
I don't know if you can get microbiome data from paleontology unless you find some
No.
No, it's more.
from
It's more. It's more about the approach that they took, like the AI. Like I'm curious to know, like in you know, and basically in his studies in his field, how are they using, um, like Ricardo, are you guys starting to use, you know, any kind of artificial intelligence algorithms, loading up the data and start to look at it, you know, in that sense, just because, you know, that's been very interesting.
The
I
only
was in the,
stuff
yeah.
that I've seen really, it's for like helping with segmentation. That is to say, like okay, we do scan these fossils in like computed tomography machines, like your regular CT scans. When you break your bone, you just go to a medical facility and you scan the bone and you see where the fracture is. We use the same thing, just like on steroids. Basically, we go to the synchrotron, which is like you know, an extremely powerful CT machine, or we have lab computed tomography machines, which are just not as great, but they do a lot of good stuff as well. So yeah, so when you scan a specimen, you can basically teach the algorithm to learn how to separate bone and rock. So that's the only really kind of applications of AI I've seen to paleontology. And there's a software called Dragonfly that does that. But I've never tried it before. I prefer still to rely on my own senses in terms of like separating these bonds, because it's really tricky. It's really tricky. Yeah.
Yeah, I think you have to have a lot of data, right? And you probably don't have too many samples. Like the sample size is probably not, in the amount that it would make sense.
The amount of data generated by a computer tomography machine is really huge. The problem is to actually be able to see the sutures between the bones, because they are often co-ossified, the bones. You have to have oftentimes some kind of like a priori knowledge of where the sutures are going to be, and then there's like fractures. You know, how can you really teach a machine to separate what a fracture is and then what a suture is? It's really tricky. But I think I've seen people successfully using AI to separate the bones. It's not so much about the amount of data; it's about really the quality of the data that you have, or that computer tomography machines can generate from fossils. If you would do it with like extant bones that are not deformed, then that's like easy. Any, I bet if you put it in Dragonfly, you can separate easily the bones. Yeah, but I've never seen any other kinds of applications of AI to paleontology.
No, the only thing I recently read was, like, not paleontology, it was archaeology, to kind of go through satellite data and like find spots where you could maybe find something.
Ah, yes. Hmm.
Like, yeah, that was recently what I found, but other than that, but I'm not an expert, so who cares? Like,
No, yeah, I've seen that too for paleo. Now that you're talking about it, not sure if it was really AI, but yeah, maybe it was. But I've seen something like that. To like first, yeah, to just kind of like have an idea of where to go, really, to find anything of interest.
I don't
yeah,
think I caught this
exactly.
earlier, which was just real quick. How did you get into, like, how did you pinpoint this part of the ear to use as a marker to study? Was that something that was there before? Or like, how did that idea come to you, or the thought, or the process? Like, it's a very very nichey little, even down to the to the specific bone, right? Like, I wonder how'd you get there?
So I mean, as soon as paleontologists got their hands on a CT machine, they of course they threw as many fossils as they can to the CT machine, and they start to explore basically new parts of the anatomy that were not well known. I guess I was kind of like caught in that wave. So the one of the easy things to actually segment. So when I say segment, is like to extract from the scan. Um, because a scan is like a series of slices, and you say segment because each slice is like a segment, and you extract that segment from the overall scan. So one of the first things that paleontologists started to segment was the inner ear, because that was kind of like easy to see in the scans, because it's basically you have bone, and then you have the inner ear is like there's no bone, so it's the density contrast is really big, so you can see that quite clearly. uh, so that was like one of the first things that people started to segment and to make renders in 3D. So I was kind of like caught in that wave, and when I started to accumulate data on this, I started to notice like really some striking differences between the morphology of the inner ear in these mammal ancestors that we were finding in Mozambique, comparing to mammals. And these mammal ancestors had more like of a reptilian shape and size compared to mammals. Mammals have like if you show me a inner ear of any mammal, I can tell you immediately this is going to be a mammal, this is going to be a bird or whatever, because the morphology it's very specific for each one of the groups. But the morphology for mammals, it's really unique. It's like the canals, they are called semicircular. They are really circular. In mammals, they are really thin and they are really small, comparatively speaking, when you scale for the body mass. Okay, when you look at mammal ancestors, and when I started to look at this stuff, I started to see that the size of the inner ears were really large, much thicker comparatively speaking to those of mammals, and eccentric. They were like more like of ellipsoids than really circles. Although the name semicircular canals, despite the name of semicircular canals. So I was started to wonder why was there this difference. So eventually, I started to talk to a colleague and now friend of mine who is also the first author of the paper, Romain David, who's a French guy working uh, in ear in paleontology as well, and we started to discuss and really wonder what these differences were. And he had more like of how do you say, a more technical approach to this. And he started to say, hey, have you noticed at these equations, if you flip around a few things, uh, maybe these differences that we are seeing have some impact on the semicircular duct biomechanics. And we then started to explore this. And that's when the idea came about using the measurements and these equations that govern the biomechanics of the semicircular canals, to use it as a proxy for temperature, because we noticed that temperature was within these equations. So it's like getting a thermometer without a thermometer. So yeah, we 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 explored that idea further by looking at the fossils, by looking at the extant mammals and so on, to start working this out. So yeah, and it worked. That's why it worked. Resulted in this paper.
So last question is, which mammals, like which species in the mammals have the most sophisticated hearing, and maybe what was most interesting to you, like you're probably doing studies like, wow, this is very unique and different and interesting, just interesting information, I guess. Which one has the most sophisticated, wide range, or whatever species that you've seen, and whatever other like what's the most kind of interesting thing that popped out in your studies thus far around mammals and ears?
Not I cannot speak so much for the hearing, because what really all right, the vestibular organ, or rather the inner ear, detects two things. Although it's called the ear, it detects two things. It detects the sounds. That's the cochlear system. That's that coiled stuff that in the part of like it's the ventral part of the inner ear system. The dorsal part of the inner system is the vestibular organ, which detects angular accelerations of the head. Okay, there's you know there's not so much sophistication that goes into that part, to be honest, because it's really just it's this as I mean, the best sophistication that you have is the same as the locomotion that you are dealing with is highest. Okay, so the faster the animal, the more sensitive your system needs to be optimized for that speed and for those movements, right? So it's not so much of a sophistication. It's like nobody's talking about the accelerators on your cell phone, because your cell phone has the accelerators as well, because it's like the probably the less interesting thing about your cell phone is like when you rotate it, it turns on the lights, right? It's like, so there's no much sophistication that can go into that. What something that though surprised me when we were dealing with this is the fact that there are ectothermic, so cold-blooded mammals like the naked mole rat. I don't know if you guys ever heard of this creature. It's not so much related to the inner ear system, but it's something that really shocked me.
What's that called? I'll put up a link. Naked mole. What?
It's the ugliest creature ever. It's the naked mole rat.
Okay, the ugliest creature ever. Hold on, I gotta look that up. I'll
No,
put.
you gotta look at it, and please confirm if it is not ugly.
It's kind of cute.
Okay.
Yeah, very little.
Yes. So
Something mole rat. Wait, which one? What was the first word?
it's naked, naked, literally
Mole
as
rat.
someone without clothes.
Got it.
Seem like dangerous search terms to me.
It's in the chat, like the search for it. Yeah.
So yes, the naked mole rat is actually a rare example of an ectothermic mammal. So mammals have been warm-blooded. We found since 233 million years ago, and there's an extant mammal that's like against all odds. It's actually ectothermic. So they do produce some internal heat, but the internal heat still it's not enough to make it correlate uh, vastly with the external temperature. So if you put a naked mole rat outside of its burrows, it's likely to die. So yeah, that kind of really surprised me. That evolution sometimes can be not so reversible, but so dynamic, because as I said, the naked mole rat still can produce its own internal body temperature. For example, the naked mole rat has BAT, so that's brown adipose tissue, and brown adipose tissue is just an adipose tissue that its entire function is to produce heat to the animal, but still not in enough quantities to make it uh, vary its temperature strongly with the environment, just as any good old lizard or turtle or something of that sort. So yeah, that's one of the example, and another example is Xerus spermophilus. That's kind of like just like a random squirrel that's apparently able to fluctuate its body temperature as well. I was really surprised to know that there were still in the mammal realm these kind of creatures that can vary their body temperature immensely. So yeah, that was quite interesting.
When you say mammals have been endothermic since, does that mean there was a time when mammals were ectothermic?
That's a good question. Yeah, that's actually a little bit of a mistake that I did there. No, mammals have always been endothermic. It's in the clade of Mammalia morpha, so preceding the origination of mammals. that it's calibrated at 233 million years ago, that became endotherm. So everything synapsid non-mammalian morph is ectothermic, right? So all these animals preceding, so that they are in the evolutionary lineage of mammals, but they are not mammals yet. They are they are synapsid non-mammalian morphs. All these animals were ectotherm, yes. So we
And
had ectothermic ancestors, indeed, cold-blooded ancestors.
and can you tell us more about the species fossil that you discovered, and what role does it play in your correlation between the vestibular viscosity and the difference
The
between
one I
the
the one I found in Mozambique.
yeah.
Okay, yeah. So that belongs to a very well-known group of mammal ancestors. Oh, I mean, we don't really use the term in paleontology. We don't really use the term mammal ancestors. We just use synapsids because that's the clade that includes mammals and includes all these other creatures that are in the mammalian lineage. And it's just really the better term to use. It's more phylogenetically comprehensive, basically. So this Nyasodon fumukazi that we found belongs to a group of synapsids called dicynodons. Okay, and these dicynodons we found, and their eating ear clear reveals that they are ectothermic. Every single one of the dicynodons that we found give a signal that they were all ectothermic according to our metric. So yes, all these animals around this time. So dicynodons, they lived from about 260 something million years ago to about 230, and during their existence they have always been ectothermic.
And then what's the discovery? What's the difference in the one that you discovered? What's the what makes it different?
It's not. It's not. I mean, our paper is not so much about a particular discovery. It's about the general evolution of the group, basically. So we find. I mean, our fossil in the one we find in Mozambique just fits perfectly the pattern of all other dicynodons that have ever been found in the world. Okay, in Zambia, in South Africa, in Russia. So it fits perfectly that one. I mean, I just mentioned that one because that was kind of like one of the fossils that started making me think about this stuff, basically. Because we scanned that one and it had a nice inner ear. It had everything preserved. We isolated every single bone of it, but it just fits the pattern as expected. No crazy things going on with Nyasodon in Mozambique.
Okay, and just one more quick question: is there any correlation between the speed of movement of an
Mhm.
animal and the vestibular fluid viscosity?
That's actually one of the really interesting phylogenetic generalized least squares correlations that we did in our. I mean, that's a fancy word to say a phylogenetically corrected correlation. Um, that we found that the semicircular duct function does correlate with maximum anaerobic speed. That is to say, the maximum speed that an animal can do for a short burst of time. So yes, there is correlation of both metrics of semicircular duct function. So upper corner frequency, both relevant metrics, upper corner frequency and sensitivity of the semicircular duct function with maximum anaerobic speeds, and for that matter, we also compile the data for maximum aerobic speeds, and we do find a correlation with that too.
Interesting.
So
Thank you.
in other ways, yeah, the if you go to the supplements of the paper, this might be interesting for the person. I'm sorry, I did not memorize the name that was working on the cochlear stuff as well. But yeah, we did explore this a lot in the paper.
Yeah, thank you for answering those questions. My kids will run into the car in a few minutes, so I will be loud in a few minutes. But wanted to ask a question more related to, you know, the institute, the lab you built, because I know there are a lot of people here and on Clubhouse that kind of, you know, plan or do similar stuff. If it's an institute or, you know, a research company and so on, like, what is the advice you can give people that want to build something like this in a very remote area with no
Hmm.
budgets? Like how did it work? Like, how did you succeed, and what's the advice you could give people?
Find the right person to work with you. That's really the only good advice I can give. Uh, because if you don't, we were lucky enough to when I started to go to Mozambique to and starting this project. There was this guy called Luis Costa Jr. Unfortunately, he died on a car crash a few years ago, and he was amazing. He was just a facilitator. He just made everything easy in like a country that was not ours, and he saw the potential of the project, and now we are kind of like collecting the fruits of this effort. And it was really a bilateral collaboration. These things cannot be done, just by my strong will or whatever. These things need to be done with a team of committed people that see the potential and can really raise the resources for something to happen, because otherwise it's just like it would feel like swimming against the current. So the key is people. Find the right person, and and really work those relationships. I think that's what really matters, I would say.
Yeah, thank you for that insight. That's really interesting.
Mm,
Um, yeah, it's really an important insight. And so yeah, I would say congratulations for everything you did, and we wish you all the best, all the luck,
mhm.
all the funding, and that,
All
you know,
right.
you continue to have a lot of fruitful work and have a lot of
Mhm.
students. And yeah, I think, you know, it's really amazing what you're doing, what you did. And yeah, we were very honored to have had you here. And I wish you all I think we all wish you all the best for the future, and maybe you come back one day, maybe
Mhm.
also with a student or so, to talk about new or further projects further along in the future. Thank you.
Yes. Thank you so much for having me and letting me speak for so long. And I'm sorry for the noise.
Oh yeah,
I
no.
bet you heard some heavy music.
Oh, don't worry. On Clubhouse, it's supposed to be informal. And no, like, thank you for
Yeah,
taking
and
so
feel
much
free
time.
to invite me. This was fun. Honestly, I enjoyed it. Of course, I mean, letting a scientist or a paleontologist speak about its work, that we want no more than that, honestly. So it's a lot of fun. I always love to talk about what we do and our discoveries. So it's fun. Thank you for listening to me and for all the questions.
Yeah, thank you. Yeah, come back anytime. Like, all I can say.
Okie doke.
Like, anytime.
Okay, bye bye. Thank you.
Okay. Have a good weekend. Enjoy Portugal. I miss it so much. Okay.
Ciao,
Okay.
ciao. Ciao.
Ciao.
Bye.
Ciao. And thanks everyone for coming and for discussing and commenting and all the questions. And enjoy your weekend. Happy Friday. And yeah, thank you. We have more. Oh, right. Some advertisement. We have more rooms like this next week again. We will have Dr. Lee. He will talk about first solid state light driven nanomotors. It will be really interesting nanotechnology research that he will share with us. Then on Tuesday, Dr. Lafay will talk about thymocyte cell type discovery and leukemia. It's really he made really breakthroughs in leukemia research. And on Wednesday we will have an AI room that diagnoses dementia accurately and earlier on. Thursday, an ETA2 that will be able to continuously measure blood pressure, which is like one of the most important data points to for health, like long-term health, is blood pressure monitoring around the clock. And yeah, as we learned in the sleeping room, really around the clock and not just acutely at some time points during the day, also at night. So this ETA2 could be, the technology of the future that we will need. And yeah, and okay, so hear you all back soon. Thanks for coming and enjoy your weekend. Thanks, everyone. And close the room in 3, 2, 1. Bye, everyone.
Bye. Thanks.
Thank you.